So girls, today we will start with the new topic, that is the achievements in crop biotechnology. Now, what do we mean by biotechnology? Biotechnology, it includes a wide range of technologies, okay, or a wide range of procedures for modifying the living organisms. Now you have already studied about genetic engineering, where we can modify the living organisms both for both plants and animals, and even the microorganisms according to human purposes. So genetic engineering is nothing but it is biotechnology. And you have also studied with Miss Rimea the plant breathing. Plant breathing also is one of the methods of biotechnology. Okay, it is actually the first method, one of the first form of biotechnology, plant breathing. Okay, where we have seen that the farmers they have altered the genetics of their crops. How? Through introducing them to new environments and breeding them with other crops. So plant breeding also is a form of biotechnology, and even genetic engineering and any other techniques or procedures where the living organism is being modified, okay, according to the human purpose. So but today we are going to talk, we have already studied in genetic engineering, where it has a wide variety of applications in for example in medicine, in agriculture, in industry. Okay, it has a wide variety of application, but in this topic we are going to talk only about crop biotechnology. That is, what are the achievements of these biotechnology procedures or um, biotechnology procedures for improving the crop plants. Okay, that means we will talk about the agricultural biotechnology. Okay, these points here I've written, okay, which highlight the achievements of biotechnology in crop improvement. Okay, so the first one here, as you can see, it is herbicide tolerance crop plants. Now, these development of the herbicide tolerance crop plants, it was actually the first major achievement in crop biotechnology. Okay, employing the genetic transformation technique. Okay, through the use of genetic transformation or genetic engineering, these herbicide tolerance crop plants, they have been developed. Now, transgenic plants, okay, that means those plants by which uh, their genes have been modified, those are called transgenic plants. All right, so these transgenic plants resistance to a number of herbicides, they have been successfully developed. So this is one of the achievement in crop biotechnology. And these plants, they are able to resist to a number of the herbicides such as imidazoline, then glufosinate, all these herbicides, okay, which sorry, these plants which can tolerate these herbicides can have been successfully developed. Now, we know that herbicides, they are mostly used in agriculture to control the weeds, the unwanted plants, that is the weeds. But then attention has been given to during recent years for the production of the herbicides which are less harmful to the environment. Okay, for production of safe and biodegradable herbicides. But these new type of herbicides, they actually harm the plant, okay, by disturbing the process such as biosynthesis of amino acids and photosynthesis. So these herbicides, enter the plant, they affect the plant and disturb these natural processes such as biosynthesis of amino acids and photosynthesis in the plants. Therefore, due to the development of these new herbicides, it has become necessary to produce such plants which can tolerate or which can resist these herbicides. So this is one of the major achievement in of biotechnology for crop improvement. Now the second one is the engineering for virus resistance. This is another major achievement in crop biotechnology where various biotechnology technological attempts have been made to achieve resistance for pathogenic viruses in plants. We know that plants they have been attacked by various pathogenic microorganisms such as fungi, bacteria and viruses. So attempt has been made to achieve resistance to virus, okay, to pathogenic virus. Now transgenic tobacco as well as tomato plants they have been obtained, okay, have been successfully obtained using the coat protein approach, which are resistant to tobacco mosaic virus and tomato mosaic virus. Now, similarly, all other transgenic plants they have been achieved having resistance against other viruses such as alfalfa mosaic virus, cucumber virus, and so many other pathogenic virus. So these plants they have been successfully um, produced. Okay, the plants which are resistant to virus. So now I'm going to talk about our next major achievement, which is also mentioned in your syllabus, resistant to insect attack. Okay, the crop plants which are resistant to insect attack have been successfully um, produced. And one of the major example is BT cotton. Okay, so let us talk about this. What is this BT? Now, BT here it comes from the name, this term it comes from the name of a microorganism, a bacteria that is Bacillus thuringiensis. Okay, this is the bacteria. Now this bacteria Bacillus thuringiensis it has a gene, okay, named as BT gene. Okay, after its name only BT gene, or also referred to as the cry gene. Now, what actually this cry gene it does, okay, we know that from a gene, from a DNA, transcription and translation will take place, right? Transcription and translation, and after transcription and translation takes place, then we will get the proteins. So these proteins they are referred to as the cry proteins. Okay, so why the name cry proteins? Because these proteins they are crystalline in nature. So crystalline proteins. So these cry proteins actually they get dissolved in alkaline pH. And when they get dissolved in alkaline pH, they become toxic. Okay, so they become toxic. But what happened? This cry proteins which are produced inside this microorganism, that is Bacillus thuringiensis, they dissolve in alkaline pH. But in plants, okay, the pH is not alkaline, okay, in these plants. So what the scientist does, they actually isolate this gene from this Bacillus thuringiensis and introduce this BT gene into the plant. Okay, this cry gene, it has been introduced into the plant genome. Now, we know that when this gene it has been introduced inside the, of course, how it introduced, then you have already studied genetic engineering and gene cloning. You will understand with the help of the vectors, they will introduce these genes into the plants. Now, when they introduce into the plant, these they will undergo transcription and translation again, then they will form the cry proteins. Okay, so these cry proteins they only dissolve in alkaline pH, but the pH in the plant is not alkaline. Okay, so they remain as it is. They will not get dissolved. They remain in a crystalline form. But when a larva, okay, of any insect, the larva of insect, uh, it feed on this plant, suppose in the leaf. Okay, it feed on the leaf of the plant. What happened? This cry protein it gets into the gut of the pests. Okay, it gets into the guts of the pets. So here when it gets into the guts of the pets, it binds to the epithelial cells of the gut. When it binds to the epithelial cells, what happen? It will form perforations. Okay, it will form perforations on the wall of the cells. Now, due to the formation of the perforations, when the water it will enter through these perforations on the cells, okay, of the alimentary canal of the pests, then the cell it will swell due to the when the water enters inside, the cell will swell and it will ultimately ruptures. So it will finally kill the pests. So we see that how these cry genes from this Bacillus thuringiensis, it has been transferred to various crop plants. Actually, not only cotton, where you have heard this name BT cotton, but so many other plants like BT brinjal, uh, BT tobacco also it has been successfully produced, and many other plants also. But this BT cotton, it has been introduced in India since the year 2002, and till date we are using this BT cotton. Okay, so you can see that this also is one of the major achievement in crop biotechnology. So next is the resistance to pathogenic fungi and bacteria. Now the genetic transformation technique or the biotechnological attempts has been also made to produce the transgenic crop plants which are resistant to pathogenic fungi and bacteria. Okay, these we know that there are various losses in agriculture due to the reduction in crop yield because of these uh, fungal and bacterial infection, fungal and bacterial diseases in the plants. So this can be prevented to some extent by cross protection of plants from the pathogenic fungi and bacteria. And the next one we have engineering for tolerance against abiotic stress. So abiotic stress like drought, flood, salinity, heat shock also can be resisted by using transgenic plants. All these abiotic stress, we know that they affect the growth of plants and thus they cause a major loss in agriculture, right? So to prevent such losses, these genetic engineers, they have developed the stress tolerance plants of a number of crops, not only a single plant, but of various numbers of crops. And for this purpose, several plant genes they have been cloned, which encode for enzymes that are very important for avoiding loss due to different stress. And the next achievement is the introduction of male sterility in crop plants. So We know that male sterility, those male sterile plants, they use to produce non-functional pollen grains, right? They are not able to produce functional pollen grains, thereby prohibiting fertilization of the ovum. Okay, they do produce viable eggs, but they are not able to produce functional pollen grain. So what is the use of this male sterility? Now, male sterile plants, they are advantage for the plant breeders. Okay, why? Because the cross between the two genetically different lines, it will produce the hybrid seeds. Okay, so to facilitate the production of hybrid seeds, uh, male sterility is important so as to produce these hybrid seeds, and such seeds, they usually results in larger and more vigorous plants. So that is why it is very much it is a big advantage for the farmers, okay, for the plant breeders. Now, some male sterility causing genes, okay, those genes which cause male sterility, they have been introduced to various crop plants, okay, to engineer male sterility in plants like tomato, then maize, cotton, oilseed plants, etc. So these genes which cause male sterility in the plants, they have been introduced to various other plants so that they can achieve this production of hybrid seeds, because we know that seeds, they are usually more vigorous, okay, the hybrid seeds. And example of one of the hybrid, sorry, not hybrid, I mean the gene which is responsible for causing male sterility in plants, okay, for male sterility in plants is Barnase. Okay, this Barnase is actually obtained from, again, from a bacteria, Bacillus amyloliquefaciens. Okay, this gene, it has been obtained from this microorganism, and so many other genes like role B, then role C, and other genes. These genes, they have been introduced to the plants, and they are able to induce male sterility in plants. But you will see in the notes I have given, there is one gene. The name of the gene is bar star. This bar star, actually, it is the inhibitor. Okay, it usually inhibit Barnase. Now, because it is the inhibitor of Barnase, it is an effective fertility restoration gene. This is the restoration gene, fertility restoration gene. So, when they want to restore the male fertility in the plants, they can use this bar star, because what this bar star gene, it does, it inhibits Barnase enzyme. Okay, so the fertility of the plant can be restored using the gene which produce this bar star enzyme. So we see that another very important achievement is the development of transgenic plants which has a high quality improvement. This is another uh, point which is mentioned in the syllabus about you must have heard about this golden rice. So let us talk about this golden rice. So when we talk about this development of transgenic plants with quality improvement, the best example is golden rice. Excuse me. This golden rice, actually, it is very rich in beta carotene. beta carotene is like a raw material for vitamin A. So it is a precursor of vitamin A. So why? Because scientists, they know that rice is the staple food in many countries, and there is a very much deficiency in vitamin A. Okay, vitamin A deficiency disease. They can, so that is why they produce these golden rice by incorporating the gene for beta carotene into the rice. Now, why this rice is referred to as a golden rice? Because of this beta carotene. Due to the presence of this beta carotene, it imparts a golden color to the rice. So that means a gene has been introduced which into the golden rice, which produce this beta carotene, and beta carotene, from beta carotene we will get vitamin A. So this problem, it has been solved, okay, due to the production of this golden rice for obtaining uh, vitamin A. So these, just remember that the seeds of these transgenic rice, they are yellow in color due to the presence of this pro-vitamin A or beta carotene. Now the rice, the work is also done in India, okay, by the scientist Ashish Datta in the year 1992 to 2000 for the introduction of amal gene. Okay, this amalgene has been introduced from amaranthus. It has been introduced from amaranthus into potato. So this, it holds the promise for enhancing the nutritional value of low protein food. And these transgenic potatoes having the amal genes, they are undergoing field trials, and actually this is the work done by the Indian scientist itself, that is Ashish Datta, in the year 1992 to 2000. Okay, so uh, we see that the quality improvement, these are the examples where the quality improvement has been achieved in crop plants, okay, in transgenic plants. And the next one is the nitrogen fixing ability. Now you have studied about this nitrogen fixation, and you have studied that the plants such as rhizobium, then Klebsiella, and many other microorganisms, also some of the algae, the blue-green algae, they are able to fix the nitrogen. We know that the plants, they cannot use the nitrogen directly from the atmosphere. After these microorganisms, they fix this nitrogen and convert it into simpler forms such as ammonia and other forms which can be used by the plants. So these microorganisms, they are important. So these Klebsiella, then blue-green algae and other microorganism or the bacteria like azotobacter, also these are the free living bacteria which can fix nitrogen. But you have studied that these rhizobium, they form a symbiotic relationship with the roots, and they are able to form the root nodules in some leguminous plants. So these, all these bacteria, both the free living and the symbiotic bacteria, they contain this NIF genes, okay, in their genome. This NIF genes, they will produce those enzymes. If you remember, the one responsible for fixing nitrogen are the nitrogenase enzymes. Those enzymes are being synthesized from this NIF genes. So it is believed that these NIF genes, they can be cloned in the crop plants, and when they are cloned in the crop plants, so the need to use fertilizers for enriching the soil with nitrogen, it can be eliminated, because if you introduce these in the plants, then the plants themselves, they can fix the nitrogen, so there will be no need for giving the fertilizers rich in nitrogen. Now efforts are being made in this direction to transfer the NIF genes, okay, this NIF gene clusters from bacterial or cyanobacterial strains to the plants. Okay, so efforts are being made, and so far the success has been achieved, okay, but only in case of sugarcane and wheat. Success has been achieved only in case of cane and sugarcane and wheat. In these two plants, the improvement for NIF genes has been transferred successfully transferred into the genome of these plants, but research is still going on, is still being carried on for other plant species. Okay, so we see here the next one. Here the next achievement is production of novel substances in crop plants. So in this case, biotechnology is finding a novel uses beyond food. Okay, we know that crop plants, we mean we always mean for the production of food, but the biotechnology is finding the novel uses beyond food. For example, oil seed they can be modified to produce fatty acids for detergents. This is one of the example in biotechnology where the oil seeds, they are modified for production of fatty acids for detergents, and potatoes, tomatoes, rice, tobacco, lettuce, and other plants, they have been genetically engineered to produce these insulin. Okay, they have been genetically engineered to produce insulin and certain other vaccines. In future, advantages of these edible vaccines would be enormous. Okay, so we can just consume these vaccines by through consuming these genetically modified crop plants. And also another achievement is the production of useful byproducts such as drugs and materials. Now in case of drugs, the bananas, they have been successfully developed to produce human vaccines against infectious diseases, okay, such as hepatitis B. Now tobacco plant also, they have been developed that produce the therapeutic antibodies, sorry. So these crop plants, they are also they have been modified in order to produce useful byproducts such as vaccines, okay, for human, for to fight against infectious diseases, and also for the production of materials. Now several companies and labs, they are working on engineering plants, on modifying the plants through genetic engineering that can be used to make bioplastics. Okay, that work is still going on to make bioplastics. Now potatoes also, they are produced more industrially useful starch have been developed as well. So we see that these are the major achievements of biotechnology in agriculture, or agricultural biotechnology.